Multiplexed, high-throughput flow cytometry has been used in cell-based and molecular assays for drug discovery and repurposing. Today, I would like to describe how it has been used in discovery and characterization of small molecule regulators of low molecular weight GTPases. Flow cytometry is a widely used clinical and research tool that has applications for molecular targets as well as suspension cells. In high throughput applications, these could include immune cells, cancer cells, bacteria, fungus, and host pathogen interactions. By overcoming the limitations due to manual sample handling, high throughput flow cytometry could be used to screen single or multiplex targets for drug discovery, drug repurposing, personalized medicine, monoclonal antibody discovery, or immuno-oncology. It is already well appreciated that the multi-parameter aspects of flow cytometry can be used in multiplex format for the evaluation of multiple targets simultaneously. An important feature of flow cytometry is the potential for discriminating free and bound fluorescent probe without a wash step. As a cell or particle transits the laser beam, the signal detected may be comparable up to defined concentrations with or without unbound ligand. The HyperSite high-throughput flow cytometry platform delivers samples using an autosampler connected to a flow cytometer. It inserts air bubbles between samples from multi-well plates. When 4 cytometers are run simultaneously in a quad format, as described by Bruce Edwards, it allows sampling of up to 1,536 wells in 10 minutes. The technology is supported by more than a dozen patents. The IQ is now available worldwide through Sartorius. Bruce Edwards has also developed software for synergy analysis of drug combinations by flow cytometry, which was featured in a special edition of SLAS Discovery for high-throughput flow cytometry that he edited. A signature project at UNM involves multiplex analysis of glutathione transferase or GST-tagged GTPases bound to glutathione microspheres. The project has led to several dozen publications and patents, as well as clinical trials. Given the possibility of kinases as drug targets, we wanted to identify small molecule regulators of GTPases, which were thought to be undruggable. Kinases and GTPases have similar as well as distinct features with respect to binding site and regulatory protein site interactions. However, GTPases have been thought to be undruggable because of the apparent lack of binding pockets other than the nucleotide binding site. There is, however, a regulatory site associated with the binding of effector proteins. Low molecular weight GTPases consist of a superfamily of about 150 proteins that are involved in receptor signaling, membrane trafficking, cytoskeleton, nuclear import, and among other processes. In this regard, I would like to call your attention to the NIH initiative called Illuminating the Druggable Genome. I show here that you can link the importance of targets based on their publication record, in this case, GTPases, to their novelty and relevance to particular diseases. I call your attention to these websites, newdrugtargets.org and pharos.nih.gov, which can help you explore your own targets of interest. Low molecular weight GTPases act as molecular switches in the exchange of GDP for GTP. The switches are regulated by effector proteins that can be either activators of cleavage of GTP or release of GDP. As you will see shortly, the effector proteins may also be used in assemblies with the GTPases to evaluate the mechanism of action of small molecules. We have used a multiplex approach to search for small molecules that regulate the molecular weight GTPases. The GTPases are bound to glutathione microspheres that are color-coded by red fluorescence intensity. A green fluorescent GTP analog provides a binding signal that can be detected without a wash step. We conducted a screen against several 100,000 molecules in the NIH Molecular Library's small molecule repository from that time period. As shown here, both pan-activator and pan-inhibitor molecules have been further characterized with multiplexed dose-response curves. We have identified several distinct small molecule regulators for several GTPase classes. The activators stabilize GTP-bound or active GTPase conformations. The inhibitors can, in principle, stabilize GTP-bound or the nucleotide-free GTPase state in the inactive conformation. These include pan, selective, and specific regulators, which activate or inhibit multiple GTPase families, subfamilies, or individual GTPases. The mechanisms of action may be either competitive or allosteric. Shown here are examples of nonsteroidal anti-inflammatory drugs and a selective Rho family inhibitor. I'd like to spend the rest of my time highlighting several examples of small molecules. The multiplex screen revealed a selective CDC inhibitor called ML141. We show here that ML141 reduces the length of filopodia in response to bradykinin, a cell-based assay accepted as a measure of CDC42 activation, a Rho family inhibitor. Thus, the activity of ML141 in cells is consistent with its action on CDC42. The ML141 binding interaction has been characterized by kinetic analysis, here performed in bulk suspension. However, this assay can also be performed by multiplexed flow cytometry. In the suspension analysis, we follow the binding and dissociation of fluorescent nucleotide by its unquenching in bulk suspension. In flow cytometry, we discriminate free and bound without a wash step. Here, we follow the binding and dissociation continuously of the ligand in real time. We can see that in the presence of ML141, the dissociation rate of fluorescent GTP is reduced as compared to GTP-induced dissociation. In equilibrium experiments, ML141 also shifts the KD or reduces the Bmax. Taken together, these are hallmarks of an allosteric interaction. Given the role of CDC42, a member of the Rho family, in controlling cytoskeleton, we hypothesized that ML141 could inhibit cellular uptake of bacteria, as shown in a flow cytometric assay. This assay is based on the association of fluorescent bacteria with cells. The data compares uninfected cells and cells infected with bacteria in the presence or absence of ML141. The inhibition of host cell invasion by ML141 is dose-dependent and includes inhibition of uptake of methicillin-resistant strains of Staph aureus. Co-treatment with the antibiotic rifampicin with host-directed therapeutics also reduces intracellular Staph aureus infection. Further studies have shown that the mechanism is related to inhibition of actin depolymerization and adherence to fibronectin, potentially through a reduction of the affinity of alpha-5 beta-1 integrin for fibronectin, which is expected to be regulated by CDC42. In summary, ML141 is a selective allosteric regulator of CDC42. The treatment of cells with ML141 results in a number of significant cellular consequences. CDC42 may have therapeutic potential in host-directed pathogen activities, which include bacterial uptake. Others have suggested viral processing as a potential target, as well as a variety of physiological implications, which can be found by searching for CDC42 and ML141 and on PubMed. The second example involves ML282, which I showed at the outset acts like a competitive inhibitor. We can use an effector-based molecular assay to probe whether ML282 stabilizes GTPases in their active or inactive conformations. In the example shown, RILP is an effector which binds Rab7 in its active form, in its active conformation. This assay could be performed with either a Rab7 construct with GFP or a fluorescent nucleotide. As shown, when GTP gamma S is present, the complex assembles between RILP and Rab7 GFT. In the presence of ML282, there is no assembly. Therefore, ML282 is not an activator like GTP, but rather an inhibitor like GDP. Thus, ML282 is the only reported small molecule compound that shows inhibitory activity against Rab GTPases, including Rab7. It has been screened against 68 diverse targets by Recerca and shows only a 40% inhibition of 5-HT at 10 micromolar. ML282 may be used as a control compound for identifying specific GTPase inhibitors, analogous to the pan-kinase inhibitor staurosporin, used in kinase inhibitor screening campaigns. For Rab7-related diseases, ML282 provides a starting point for medicinal chemistry efforts to develop Rab7 specific inhibitors. The third example involves a large team of collaborators, some of whom are represented here. This research program is based on the repurposing of non-steroidal anti-inflammatory drugs. These studies lead to the potential of novel therapeutic reagents based on RAC1 regulation. RAC1 is also a member of the Rho family, also important in the regulation of cytoskeleton. Given that the high throughput screen identified NSAIDs as regulators of GTPases, Tudor Oprea evaluated docking models of magnesium exclusion in RAC1. Magnesium is a regulator of nucleotide binding, as well as an element of effector protein interactions. Oprea predicted that R enantiomers of NSAIDs would preferentially serve as allosteric regulators as compared to S enantiomers, which are active in the management of pain and inflammation. Tioni Baranda created GTRAP, which is a general assay platform for quantifying GTPase activation status in cells. GTRAP provides insight into the mechanism of action of small molecules in cells in vitro. In this case, effector beads can capture the active form of the GTPases. The active form of GTPase is detected by a fluorescent antibody when the effector complex with the GTPase is assembled. When R-ketorolac is made available within cells, it reduces the level of GTPase as compared to S-ketorolac. There's a variety of evidence that NSAIDs are stereoselective GTPase inhibitors. These include 1, worse survival with increased RAC based on survival data in patients in the Cancer Genome Atlas. 2, RNS enantiomers behave in an appropriate manner with respect to inhibition of GTPases as compared to COX inhibitors, which are used in pain management. 3, there's RAC target inhibition in cells, as shown by Guo. There is enhanced human survival when patients are treated with racemic ketorolac for pain management of ovarian cancer, based on Linda Cook's retrospective study. And finally, animal survival is enhanced and tumor growth is reduced with arketorolac using the model cell line of CAR8. Taken together, these observations provide a key innovation and advantage in enantiomer selective therapeutics, for which there is precedent in esomeprazole. The enantiomer of ketorolac has potential as a disease-modifying rather than cytotoxic cancer drugs, which opens possibilities for maintenance therapy. In summary, I've described several multiplexable technologies useful in small molecule discovery and characterization, as well as defining the mechanism of action of small molecule regulators of the GTPase families. These technologies have identified new chemical entities and repurposed drugs as regulators of GTPase families and subfamilies, as well as individual targets. These investigations have led to translation of repurposed drugs, a startup company for commercialization, a number of tools, and the potential for moving into KRAS family mutations as targets as well. It is worthwhile noting that by screening under multiplex conditions, information about small molecule selectivity is obtained even in the primary screening stage.